Hello, I'm Elisa Manzotti, CEO and founder of BioInsights, and a very warm welcome to the Cell and Gene Therapy Insights webinar on the latest advances in CAR-T manufacture, bridging the commercialization gaps. So as many of you will be aware, Cell and Gene Therapy Insights is very much focused on the translation of this promising new therapeutic modality, and our webinar program is central to really advancing the discussion around the critical issues facing the sector. In today's webinar, I'm absolutely delighted we're going to have the opportunity to hear from three leading experts as they share their insights into the CAR-T manufacturing pathway and how to try and navigate through clinical development through to commercial scale production. The format of the webinar today is that of a panel discussion, and I'll be posing questions to our experts over the course of the next hour. We would, as always, love to hear from our audience, so simply pop your questions in the Ask a Question box under the viewing pane, and we'll try to get to it during the allotted time. Just a little note, this is audio only, and therefore the visuals will not be changing during the session. So without further ado, I would like to introduce our panelists for today. So first up, we have Dr. Greg Rizzotti, who as Vice President of Technical Operations at Celgene Cellular Therapeutics is responsible for process development, analytical method development, clinical manufacturing, quality control, and quality operations. Prior to joining Celgene in 2006, he spent nearly 15 years at Merck, developing products that included live virus vaccines, monoclonal antibodies, recombinant vaccines, and microbially produced natural products. Our second panelist is Dr. Ian Johnston, Senior Project Manager at Miltenyi Biotec, where he is responsible for the development of cutting-edge technologies to enable cell and gene therapies to be applied routinely in the clinic. Since joining Miltenyi in 1999, he has developed a number of reagents and fully automated procedures to improve genetic modification of primary cells. And his group is also investigating new technologies to be used in CAR-T cell cancer therapies. And our third panelist is Dr. Krish Roy from Georgia Tech. Krish joined Georgia Tech in 2003, where he is currently the Robert A. Milton Chair and serves as the Director of the Marcus Center for Cell Therapy Characterization. His research interests are in the areas of scalable cell manufacturing, immunoengineering, stem cell engineering, and controlled drug and vaccine delivery technologies, with a particular focus on biomedical materials. So I think you're going to join me in thinking this is a fantastic start today, and so thank you very much for joining me today. And with that, I would like to kick off today's discussion. So one of the challenges that we often hear about in relation to the commercialization of CAR-T therapies is the issue of managing comparability and process changes as your product moves through clinical development, which can be a very short time frame. It would be great to start the discussion off by hearing the panel's perspectives on this and what approaches are being taken to address these challenges. And perhaps, Greg, if we start with you.
Sure. Hi, this is Greg. So I just would like to start by saying that it's often very challenging when you have a product that is so transformational, as are the latest CAR-T products, and has such amazing clinical results that you really want to move fast through the clinic and get to commercialization as quickly as you can. And changes invariably could slow you down. And how do you manage those without slowing the program down by getting confounding data or having to run an extra part of a trial? So I think the real key here is to understand your product and understand what makes your product work. And it's not so simple, even for a CAR-T, where we clearly know what receptor we're trying to put on our cell, but how that receptor exactly works and how it targets its antigen and how it then has this overall immunological effect on the cancer is something that's quite complex. So we need to really understand what it is about our product that keeps it safe and makes it efficacious, and as much as we can do to understand patient variables up front and then also patient responses in the clinic and having that translational data and then tying that back to the product, having good product characterization, understanding the various key attributes of our product, and then, of course, tying that back even to the process itself is very important. By doing that and having a true understanding of what makes your product safe and efficacious, then you can manage your changes better. So then when you have a change you want to make, you can then weigh the risk of the change, because invariably there'll be some sort of effect on your product characteristics. The question is, are those effects important? And then you have to weigh the risks of those differences in what the product looks like versus the need for the change. And when you're trying to move fast through the clinic, you do have the ability to, or you have the desire to not make changes, but you may want to make certain changes to make your product more commercially viable. So that's the balance you have to strike: the risk of the change versus the need for it. And the more you understand the product, the more you can assess that risk of the change. I'll pause there to see if my fellow panelists have comments.
Great. Krish, maybe we can hear from you next.
Yeah, I fully agree with Greg's response here. The way I see it is as these products develop for newer and newer indications, maybe in solid tumors in the future or larger patient populations in various cancers, sort of parallelly as we develop processes, really product understanding in terms of the critical quality attributes of the products, which we call CQAs, is absolutely critical. I think there are two aspects to comparability, right? You have the industry's internal confidence that this is a product that is going to be reproducible, that is very similar to what we had before we are making a change in the process or not. And then there is the regulatory aspect of it in convincing the regulators that you have a comparable product. And both of those can get much easier if we really know what the set of parameters of that product is that makes it functional, efficacious, safe, all of that Greg mentioned. So really thinking about what is the way the product works, how a process changes that quality of that product, so the critical process parameters on there. And as we change or tweak a process, we can measure that set of quality, and really if those set of quality that is predictive of its function remains the same, then you can solve the comparability issue. So the point is you have to do both of those parallelly as one develops the process. It's probably easier said than done, but I think that's where the field is moving along as we get into newer and newer indications. And we have a lot of tools to facilitate that right now. I mean, we really have very detailed tools to study cell properties from transcriptome to epigenome to metabolome to everything. And we really have a lot of tools in big data analytics that can help us in this process in figuring out what is the set of parameters of these cells that if I can keep constant, no matter what my process change is, then I have a high confidence that these cells are going to behave comparably as it was before. So I think implementing those early on in the process development and the product development is going to be key.
Yeah, absolutely. And Ian, any additional comments from you?
Yeah, I'd agree with both the previous comments from Greg and Chris. You certainly need to understand your product, understand what the critical quality attributes are, the characteristics of the product. It's also very important to know how you test these, and are these assays you're using to test these parameters, are they robust and reproducible and predictive of the final in vivo performance of the T cells? But looking at a maybe slightly more practical point of view, I think it also makes sense to think about how you want to manufacture your clinical product before you start any sort of critical preclinical experiments. And there I think it makes sense to make a decision on the critical components you require for the manufacture, such as the media, activation reagents, the viral vectors, things like this, and kind of nail these down quite early on. So at least these major things aren't going to change when you move later in development, because all changes early on are much easier to implement and to do comparative testing. Normally at that stage you're in vitro assays or maybe in mouse models, but you haven't taken that big plunge into the clinical manufacture. And the other thing is also for people working in the more academic research environment is also to look at which vendors you're choosing to do your experiments. So maybe find vendors who have both research and clinical grade products, so which have similar performance, so that or identical performance would be better, so that you can do your research and development activities in the research system, and then have an easy translation into the from the bench then to the clinical manufacture. And in that, you should always maybe not always think about the costs and ease of use in the R&D phase. There's sometimes a little bit of pain there to make sure that these things are compatible. So important is to think about implementing the correct processes to make sure that your product does meet these important quality criteria to ensure that easy translation from research to the clinical phase. And one easy thing, for example, is to eliminate animal components from your media at an early stage, because these can have some drastic effects then on the cell culture, the T cell phenotype, etc. So these are the things should be thought about relatively early in the development, at least after the discovery, when you're moving into the functional testing, then to adjust your research parameters to match your later downstream clinical assays.
Absolutely. And I remember sort of a couple of years ago running a webinar where we really focused on the analytical toolkit as a sort of key part of the commercialization strategy. The discussion then sort of felt, that there was still sort of lagging behind, that there wasn't sort of necessarily the right level of sort of assay development, or the assays that were being developed weren't quite helping us in terms of that speed to market. I wonder, Greg, is it your impression now that you feel that actually the sector has really made a big push around sort of sophisticating the analytical toolkit? Is this something that still has a lot of inroads to sort of really help us with commercialization?
I think it's improved on one level, but needs improvement on another. So I think in the cell viability world, if you rewind the clock 5 to 7 years ago, everyone was using trypan blue, which anyone who's worked with cells know that trypan blue is not a very good indicator of cell viability. It's one of the least stringent indicators. So having better indicators of viability and cell health, cell metabolism, cell functionality, these have come a long way. And there are a number of measures which are just much more stringent measures of viability that people now use. So that's definitely in much better shape. And then, but even there, knowing which one to use and which one's meaningful, I think you have to tie it ultimately to your potency and activity and ultimately your efficacy. And that brings me to my second point. I think in terms of potency, there's still a little ways to go to really understand how to tie your potency assay to clinical efficacy and maybe in vivo functionality. And that's something that I think every product's a little different. Everyone's got to develop that for themselves anyway. But in general, I think that's still one of the greatest challenges, because if you think about these question of making changes, if you were confident that your cell was still safe and you were confident that it was still potent and therefore efficacious, it'd be easy to make a change. But that's the one that I think still needs some work.
Great, thank you.
Yeah,
okay.
Alyssa, can I add something to that, if that's okay? Yeah, so I think Greg makes a fantastic point, and of course Ian did that too. It's essentially a lot of these assays has really improved. Again, I want to point out that viability now, if you look at viability of a cell, your trypan blue or fluorescence assays will give you a number, and if you look deeper down, say for example using an annexin type assay, you will see a lot of those cells are already apoptotic cells, and those are really not what you want. So there is really a lot of nuances, and that has come into being. But what's becoming clear is that clinical feedback back into the product quality space, and how you analyze that and how you incorporate that. It is not yet clear what the computational science behind this is when you have data from functional assay, from surface markers, from secretome, from metabolome, from transcriptome, all these variety of platforms generating data, and then you have clinical or preclinical outcome. The math for correlating the characterization data to the outcome data is not a trivial task, and we really need to develop computational tools to be able to do that. And this is not just true for cell products. You look at any other therapeutics, that's where the field is going. So the center that you mentioned, as well as this new NSF center that we were working on a lot with industry, one of the major focus is this: how you take quality attributes and then map it to clinical outcomes and then feed it back to the process and the product development cycle. And that is, I think, where we can make a lot of contribution, not only industry-specific, but pre-competitively in this space.
Yeah, that's such a great point.
Yeah, Chris, I'd definitely agree with that. And I think the leading companies now with the largest numbers of treated patients, they're in a tremendous advantage at the moment. They've got all that transcriptome data and other data that they've gathered from the patient, from the product they've manufactured at different steps during the manufacturing, and also from the patient during the patient monitoring phase. And I heard at a recent meeting that one of these companies, they've now they're processing all this data with algorithms, and they've now found predictive markers in the patient material at the beginning of the process, which, as far as I understood, then has predictive effects on understanding toxicities downstream. So you can actually predict which patients might have toxicity problems before you've even made the product, which would have ramifications then also for the inclusion criteria. Maybe at some point you can then modify the manufacturing process to make the cells less potent, or maybe to counteract the toxicities. But I certainly think that the pharma companies with this large cohorts of patients, that they're in a very good position now to find those biomarkers and develop predictive assays during the manufacturing to be able to know if either process changes or the manufacturing process itself is on track or not.
Interesting. Great. Okay, so a related aspect of sort of moving to commercial scale manufacture of CAR T therapies is how to minimize batch variation in the midst of, patient or donor variability. I was wondering how much of a challenge is this, and what approaches can be taken to try and reduce variability of your final product? Maybe Chris, if you could kick us off with this one.
Yeah, so I this is to me, and I certainly Greg and Yin can sort of lead these things every day, so they should comment as well. To me, I think it's a large challenge. Whether you are looking at an autologous product or an allogeneic product, the patient variability is huge. Certainly for our autologous product, but even when one gets to allogeneic, there will be how you select a donor issue, right? So that batch variability of the input cells is going to reflect on my output product, because in most cases, my process is constant, right? And we are yet not at a point to have a feedback-driven change in the process itself or tweaking of the process based on the initial cell characterization. And I think that's where we need to eventually get there. But I think things like, at least in the short term, really understanding the input product, again, thoroughly characterizing that, understanding what I need to have my output product characterization is, and how my process itself affects my product. So really getting into the science part of the manufacturing is going to be critical. So I want to emphasize that that's a very critical aspect of this journey. But also, tools, really. How do we measure quality or cell properties during manufacturing in an inline manner, fairly real time? And how do we take that knowledge and fit it back to the process itself, so the process engineers can figure out how do we tweak the process to ultimately affect the product quality or control the product quality, rather. And then the other thing that I think could help certainly in the variability is really automation. And not only just automation in the way we think of automation, that it's just the automation of a particular fixed process, but automation in the sense that it is feedback-controlled automation. So we understand what the measurement of the cell is during the manufacturing. We feed it back to whether it changes the feed rate, whether it changes the cytokine concentration, whatever that is. So eventually going to that flexible automation is certainly going to help. But the last thing I want to point out in that is really we cannot overemphasize the critical need of reagent quality. Cells are entirely going to be dependent upon how good our reagents are, what is the batch-to-batch variability of the reagents itself. And so in the light that our input product is quite different, we really need to think about and invest a lot of time in understanding reagent quality and sort of supply chain quality, which can then minimize some of the variations that we see in the output product. But I want to hear from Greg and Ian as well, their experience with this.
Thank you, yes. Greg, perhaps you'd like to follow up.
Yeah, so I think those are great points, Krish, and I'll just maybe expand on a couple of them. I think you mentioned having to really understand the reagent quality and really understanding how the process affects your product. So my general advice there would be, first of all, separate patient variability from process variability. Get healthy donors and really characterize as much as you can about raw materials and the product. So a couple of advantages with healthy donors. One, when you're doing experiments, you can pool them. You can pool donors and get a lot of starting materials. You have the same starting material to characterize your process. Or in some cases, you may have enough healthy donor material from one donor to go a long way as well. So you really want to eliminate initial product or initial patient variability from this exercise. And when you do that, then it becomes a very straightforward process characterization exercise that's typically done in any of these fields, in traditional biologics, for example, where you really have to tear apart the process and understand how every key important parameter, input parameter, affects your product outputs. And back to what Chris said, you need to know what the right product outputs are, because you don't want to measure everything in every experiment, because it'll just kill your resources. So certain kinds of experiments might cause you to look at certain safety characteristics. Looking at other parameters might cause you to look at potency and efficacy characteristics. And other parameters, you may want to look at everything if it's such a critical parameter. But you really need to tear that apart, understand how your inputs affect your outputs, both in terms of the process parameters you can control, as well as the raw materials and the reagents that you control. So looking at lot-to-lot variability, understanding what it is about a particular reagent that makes it good or bad. So some people call this QBD, where you look at every little piece. But I like to call it process characterization and raw material characterization, where you really just tear apart everything. So as much as people want to go to a real standard type process, I think you have to recognize that you have to pick out your parameters, what your reagents are, what your process parameters are, and then really characterize that to know what windows you can operate in, so you know what's going to work in terms of your process and reagents, and you can eliminate process variability, at least mitigate process variability as a challenge. And then once you solve that, then you can focus on patient variability, which is a whole challenge in itself.
Absolutely. Ian?
Yeah, I'd certainly agree with those comments. Maybe to just give a couple of specific examples. In the development of our own autologous CAR T-cell manufacturing process at Miltenyi, we've observed that it makes a huge difference which starting material is used. So this input variability can have a major effect. And there's many people manufacturing CAR Ts who use PBMC as a starting material. But if you look at patient material, then the proportion of T cells in that can vary from under 5% from heavily pretreated patients up to similar frequencies to healthy donors of 50% or so. So it's quite clear that if you just take those cells, put them straight into culture, you'll get huge variability in your downstream T cell activation and expansion. And many ongoing trials at the moment which are using PBMCs as a starting material, they actually have variable harvest days because the cell dose is reached on very different days. So, and that for me is a huge variable in the process. So it's a fixed process with an inbuilt variability. And we found that if you actually start with a defined cell population using enriched T cells, for example, you could remove that variability completely and have an extremely robust and tight expansion of your cells. And you remove the problems of this variable growth and problems of the viral transduction. So I think that you still, have a little bit of variation from the patient itself, but I think that will be minimized if you manage to keep the conditions the same. And another part of the variability, which Krish briefly mentioned, is also automation. because there's still a critical component of variability coming from the operator of the process. So the more manual handling steps you have in the manufacturing, or the more interventions that take place because something has to be done, the higher risk that something can go wrong, something will be mixed up, or something will be just done differently. I mean, you can have a recipe about how to make a souffle, but not everyone gets it to be nice and fluffy the same. So I think this sort of variability you get away if it's really all bits and bytes and zeros and ones, then the manufacturing is then simply more robust if you get that element away. And another thing that has to be robust are the quality assays. So you can have a robust process, but it doesn't help if your quality assays are also too variable. So you think you're measuring something, but then it's not, a fixed point. And with these CAR T products, there's, I think all of the products, there's a flow assay somewhere to look at how well the CAR is expressed, how many cells are expressing it, looking maybe at the viability as well, the phenotype of the cell. And simply gating on flow cytometry is also something which can be very operator dependent. It's like misplacing of the gate or even poor antibody staining, if the staining protocol isn't robust enough, can also lead to variations in perceived numbers of positive cells. Because these CARs aren't always expressed as a very defined population. Sometimes they're slightly more smeary. So I think it's very important here to have stringently controlled staining reagents and flow panels, which give you that robust staining performance. And also, I think we're going to see more and more solutions coming with automation of the flow acquisition, gating, and analysis, again to remove this operator variability. And also, you then reduce the workforce requirements, and we'll be moving towards also a bit more cost control.
Great, thank you. And I like the souffle analogy. I can never actually get mine to rise. But, so yeah, we've actually just had a follow-on question actually to a point that you raised there at the beginning, Krish, to this particular question. So it's coming from our audience member, and it's saying, isn't there a slight sort of risk or danger of maintaining the old sort of biologic mantra of the process is the product? And there's been such a move over the last few years to try and sort of remove this thinking from the regulatory agencies in favor of stronger arguments on process capability to handle input variability. I wondered if you wanted to respond to our audience question, Krish.
Yeah, I think you make a great point. We have sort of always had this mantra that the process is the product, but in the context of very highly variable source of material, you really have to understand the process enough to make sure you are able to tweak the process. Now, whether that is deviating from process as a product, it may not. But on the other hand, a flexible process is going to provide, in my opinion, a stronger product, or at least a more reproducible product. Because if your input, no matter how much we control reagents, how much we do automation, how much we have reproducibility of the process, if the product, if the starting product itself is highly variable, it is going to be very difficult to control the end product quality if the process remains fixed, right? So to me, at least, thinking ahead in the field, a flexible process, a flexible automation process, even if you think about automation, where the process can be intervened or the process can be augmented, the process can be improved during manufacturing based on the quality feedback of the product in real time, is probably where the future needs to go. Is that going to happen today? Absolutely not, because that's when we are not at that point in one of these products.
Oh, sorry, Krish. I think your phone is we're having a little bit of difficulty with your phone there. I know you're phoning in from India today.
Can you hear me?
Oh,
Can you hear me now?
oh yes, that's much better. Thank you.
Yeah. So what I was saying, what Greg mentioned is really teasing out the components of the process and how that affects the product quality is critical to note, but then being flexible enough of a process that allows us to really augment the process or improve the process to get to that desired product quality. Because we are going to have, age difference, large differences in ethnicity coming in as we get into the general population. The clinical trials in CAR-T are small. By any means, you would think that they have only been done in fairly small group of patients. And even the clinical indications have so far been fairly small. But as we get into larger clinical populations and get into a very broad general population across countries, across ethnicities, across age groups, genders, what chemo they got, what radiotherapy they got, we are going to have wide variety, especially in autologous products, of input product variability. So really thinking about how to have a flexible process to get to a reproducible product quality, in my opinion, is going to be key going forward.
Interesting. Thank you.
If I could just add to that, I agree with Krish that that's the ultimate state we'd like to be in, where we understand enough about what product we want to make, and we understand how the process can be modified or controlled, I should say, to adapt to the incoming material and then the product as it evolves through the process. So that would be the ideal state to be in, that we have enough control and understanding that we can always get to the same final product. And I think so in today's world, we don't quite have that yet. I think the key is to have a wide range of acceptability on your final product, as wide as you can tolerate, because different starting materials will give you a different final product. And I won't say it allows you to have a process that's out of control. I think you want to get your process in control, as I described, as we talked about the last question. But because the patient variability is in play, I think you need to understand that your product's going to look different from patient to patient, and you need to understand the widest windows you can accept on key variables. And you get that through clinical experience, sometimes out of necessity. Sometimes you have a product that maybe doesn't look like you wanted it to look. Maybe you didn't make enough cells as far as you know. But if unless you have reason to believe those won't be efficacious, you want to try it anyway and expand your windows that way. So I think that's the current state we're in. Krish is talking about the future state. We're probably going to work our way towards that state over time, but we have to be able to adapt to what we have at any particular time.
Yeah, absolutely. That's a great point, Greg.
Yeah, totally agreed.
Wonderful. Okay, so moving on to the next question. So obviously a key part of your manufacturing strategy is going to be the actual design of your facilities. Ian, maybe you could start off here on what do you sort of see as the critical considerations when designing manufacturing facilities to really meet the scalability needs for CAR-T manufacture?
Yeah, well, I think the major challenge is to make sure that you can make many products in one manufacturing room or suite. If you're wanting to scale up, you can't do this on the spur of the moment by building more clean rooms. So you need to have that ability to manufacture multiple products. And the way to do this is to use fully closed systems for manufacturing. So you can go from end to end of the process with no open steps. So every single step of the process has to be closed. Or it has to be designed to maintain this closed system by using, for example, tube-tube welding, something like these Terumo tubing welders. What this also means is that suppliers of the reagents, consumables, media, and everything else you need have to make sure that everything's provided in these correct forms so you can actually use it in the closed system. It's no use having a closed system for the manufacturing when your medium comes in a bottle. Yeah, it has to come in a bag that you can weld onto your closed system. So you need to eliminate all the isolators, all steps where you're working in an A and B environment, for example, so you can then do manufacturing in a C or a D environment. And that's where the new recommendations that the European Union have made recently for ATMP manufacture in a GMP environment suggest that a D room, so I'm not sure what it is in American conditions, I think it's 10,000, 100,000, something like that. That's fine if you're using closed systems. And with these recommendations, they also say that multiple products can, in fact, be made in one room. So for me, it's a very progressive attitude from the European authorities, very forward-thinking. And I think implementing this will really enable massive cost reductions when you're designing and building these facilities and operating them, because you don't need to invest so much in the infrastructure. And just to mention Miltenyi ourselves, we recently built a facility on these lines, allowing parallel manufacturing in one room, and received a manufacturing license from the German authorities to manufacture CAR T cells in this facility, where you can make over 20 CAR products in one room using our closed system technology. So this is something that is really implementable. It's approved, and you can actually do it. So I think that's going to, be a bit of a game changer for everyone, being able to use these closed systems to manufacture. And what we've also learned, a second point, is that the workflows that you use in the facilities have to be very carefully planned if you're making multiple products. So if you're using device-based manufacturing, so everything in one device, it's very simple because you don't need to coordinate multiple instruments during the batch productions. And there are certain days which are also extremely labor-intensive, where scheduling has to be very carefully coordinated, because if you have all your patient samples arriving or leaving on the same day, then you obviously have peak usage and things like the QC assays downstream. You'll get then bottlenecks turning up in the logistics center. So this is something that also has to be looked at in great detail. And I think the solution to this isn't just automation. The automation is absolutely critical to allow these things to work, free up the workforce. Have your clever people not working in the clean rooms, where there's an awful lot of PhDs working in these clean rooms to make sure everything's running smoothly. But, get your creative people doing creative stuff. And also use clever software solutions, automation of electronic batch files, things like this, to further reduce the burden of these scaling up with these multiple processes. And at the moment, it's hard to really conceive what there's going to be like when you're making, hundreds of batches per week. I think most people are working at the most maybe with 10 a week, the major pharma companies, or maybe 20. So the idea of scaling up to making hundreds per week and trying to get these workflows sorted, the logistics centers coping with this, no mix-ups, making sure everything's got barcodes on it, tracking the product outside, scheduling the patients, etc. I think this is there's still got to be solutions developed to be able to implement these things efficiently.
Yeah, absolutely. Chris, do you have any comments around the facility design?
Yeah, so I absolutely, it's a fantastic overview of that. I just so that.
Sorry, Chris, your phone is cutting out a little bit again. I don't know if you can.
Okay, is this better?
That is much better.
Yeah, I think I know a particular angle to hold this on now. All right. Okay, so two things I wanted to bring forward. One is this critical need for analytics during manufacturing, right? So not at the end product, and preferably not through sampling. So if we have manufacturing setups or facilities, or whether it's an automated system where the critical analytics is built into, I think that is absolutely going to be a game changer in the future, where we know how the batch is moving forward, how product quality looks, without opening the system, within the closed system manufacturing process. Whether it's a standalone benchtop system, or whether it's a centralized manufacturing facility, automated sampling, technologies, automated separation technologies, automated or real-time fast analytics technologies are going to be really critical for the manufacturing setup of the facility. The other thing maybe two things. The second thing I wanted to bring up is what I think Ian was alluding to a little bit, is the workforce side of things. I think we can build tons and tons of manufacturing facilities around the world, but we really need the appropriate level of workforce to man them, right, or to run them. And that always doesn't come from PhDs and bachelor's degrees. That at least in the United States comes mostly from two-year technical colleges. And so really running the manufacturing shifts and training those people. I'll give you an example. In my state of Georgia, in the technical college system, there is literally 3 of those technical colleges that even have a biotechnology training program, which is not going to sustain the manufacturing facilities that we are hoping to build in the cell therapy industry going forward. So really an investment on the workforce training at all levels is critical. And then this third area that I think industry is still grappling with, this whole idea of centralized versus regional manufacturing versus, on-site, bedside manufacturing, and everyone is playing differently in that space. But the point that I want to make is the critical importance of supply chain as we build, whether it's a large centralized facility or a distributed facilities around the world, or even benchtop systems across all hospitals around the world, whatever that model we end up with, the very critical need of having reagents and supply chains that are consistent between those facilities and between those centers or the manufacturing centers is going to be critical as we think about manufacturing facility design.
Absolutely. And Greg, did you have any sort of final comments around that particular topic?
Yeah, let's make 2 points. I think most points were covered. Back to workforce, because I think Ian covered really almost every other aspect of facility design. So Krish made some great points about workforce, and I think to kind of follow Ian's theme, you need a flexible workforce. So it's really challenging to know how many people to hire, because it's always tough to know what your marketing projections are going to really be like, and it would be a real shame if you can't supply the market for products like CAR-T when they really are saving lives. So you want to make sure you have enough people. If you overstaff, you're going to be really driving up your cost of goods, which again may cause patient access issues and therefore not be able to serve everybody you need to serve. So I think the way to solve that is one of the themes that Ian highlighted, which is to have flexibility. So a flexible workforce that can work on more than one product, or maybe work on more than one area in your facility, so that you really can manage the peaks and valleys of incoming patient material. Because this isn't like a typical product where you can campaign and make lots of product and get ahead of the game. You're always playing just keep up. So that's one point I wanted to make. The other point Krish made about centralized versus decentralized, I just think we shouldn't overlook the challenge of decentralization for comparability. It's going to be challenging enough to move from one facility to the next when you go into a new region. And we talked about lots of ways to mitigate the variability of the process through automation and better control. But even methods, transferring methods can be tricky. And maybe the answer is decentralization with centralized methods, for example. But I think we need to make sure that when we look at multiple centers that we have good comparability, not only when it comes to the process, but when it comes to training and how we carry things out. So I think it's something we can't minimize the importance of, is that comparability across sites will be something that we have to pay a lot of attention to, put a lot of resources on. So how thin do you really want to spread yourself is something I would just warn people.
Yeah, excellent point. And so, really sort of building upon that, obviously as these transformative therapies start to be utilized for larger disease indications with potentially hundreds of thousands of patients, that supply chain is going to become even more critical and more complex. I think if we sort of dig a little bit deeper in that, what do you sort of see as those specific challenges associated with the CAR-T supply chain, and sort of what kind of steps can be taken to try and mitigate those risks as we move to this larger scale manufacture? Maybe Krish, if you could start us off with this.
Yeah, I think we discussed quite a bit of supply chain issues already, so I'll kind of keep it short here. My thought process in the supply chain, as we really go into this 100,000, 200,000 patient populations or even more, is sort of ensuring redundancy in the supply chain and robustness in the supply chain. If you look at any traditional manufacturing industry, there is a lot of built-in redundancy in the supply chain. Even then, you hear, the small plant in Michigan stopping for a few weeks and GM has to stop making car productions, right? That happens very rarely, but it still happens. And the emerging industry like cell manufacturing or cell therapy is extremely prone to that because many cases, our reagents almost dictates the quality of the product. And we are very much dependent a lot of the time on single supplier basis. Now, one can hoard, tons and tons of supplies, but that again increases cost of goods, and then these have specific shelf lives and so on. So it's a very difficult balance that one has to strike between primarily single supplier basis, single reagent suppliers, maybe two, and it's very little robustness and redundancy right now. If you do some modeling and if you think that, if a supplier goes down and if I have X number of patients that I need to serve, and these are critical patients, some of them can live 3 months, some of them can live 5 months, so we cannot really have a production stoppage for too long. It's a very critical issue that has, in my opinion, very understudied because there are a lot of tools that are already available and knowledge in the other industry on how supply chain disruption affects product output. You will see that, really a center can be decommissioned, or it may take months, if not years, to come back up to steady state, even with a single supplier disruption. And those type of models really, in my opinion, need to be studied very detailed to think about what kind of reagent redundancy and supplier redundancy needs to be built in. And that's on sort of the reagent front end. And then obviously the logistics and distribution end of the product is another issue. And there's certainly a lot of innovations that needs to be there. But I want to hear from Greg and Ian as well on their experience on the supply chain side.
That's Ian, perhaps you could follow on?
Yeah, and it's certainly a major issue. And one thing about the maintaining the quality of the products for the customers. Often customers use products for different uses than the one that the manufacturer has tested. So the question is, does the manufacturer have a quality test for his product, whether it's a medium or some other reagent, which would be predictive of changes then for the use the customer is having? So if you're using a general medium for T-cell expansion, for example, and it's tested in quality on 2 or 3 cell lines, and maybe 1 or 2 primary cells, but in a different sort of assay, will that manufacturer be able to pick up on the changes in the production variability that might have an effect on your process downstream? So I think that's something where you probably have to have quality tests on new batches of certain things if you're using the product in a slightly different way, maybe from the original planned use of the manufacturer. And from the manufacturer side, it's obviously critical to correctly predict the customer demand and be able to react accordingly. So to avoid running out of stock, most large volume users of products will sign supply agreements with the manufacturers with detailed forecasts so they get these products. But as Greg said, it's also very difficult to know, how many patients you're going to treat. Is the recruitment going to be on track or not? So this demand planning from the manufacturer side has to be such that they have a good predictive system for the number of patients, but also that they order reagents on time to have them at the manufacturing site to be manufactured. So you don't want to miss ordering something on time If there's maybe a 2-week delivery time, you have to make sure you deliver well in advance. And I think here also some degree of automation using integrated software solutions to manage your inventory, and the shipment is going to be key. I think there's a number of manufacturers and a number of CAR-T manufacturers who are also looking at this area, trying to work out how to control and coordinate their supply chain to make sure that they always have everything on stock in the right spot, and also, of course, supply chain in the sense of making sure that the patient materials and logistics part is well coordinated, and that these other aspects are looked at. So, and that's the second point is also integration. So I think you need to look at an integrated approach where you don't just look at supply chain as being consumables and reagents, but it's also looking at all aspects of the patient treatment, the coordination of things like the patient recruitment, the treatment scheduling, apheresis, the apheresis transport to the manufacturing site, reagent consumable picking, so getting everything ready for your manufacturing into one spot for the batch, the manufacturing itself, the QC release, then the patient preconditioning has to be organized at the right time for when the product's delivered, and the transport of the drug product back to the clinic has to be organized as well. So I think all these different aspects, an extremely complex picture and movement of different things. This also, I think, would be best to have an integrated software solution to manage these things and to make sure that each patient gets what he needs at exactly the right time. So it's a bit like automobile manufacturing, just in time. You want to make sure everything is definitely in time and definitely there. And you can either think of one overreaching software solution or multiple separate software solutions that then interlocked in some way to manage these things. But I think as the numbers increase from tens of patients to hundreds to thousands, it's going to be impossible to keep track of all these things without some degree of software automation.
Yeah, I think that's a great point. Greg, I wasn't sure if you had any sort of additional comments around the supply chain.
Well, since Ian and Chris were so comprehensive, I'll just add one other minor point. It was a great coverage of different items. So as far as the challenge with single source reagents and certain reagents may potentially stocking out, the only thing I would advise is that the development folks really early on, as Ian pointed out earlier, when you design your process early, make good choices. The development folks should really speak to the supply chain and manufacturing experts really early on to understand where their focus needs to be. You probably can't optimize everything before you hit the clinic because you want to get to the clinic quickly, but you should really understand what the most challenging raw materials are and try to eliminate those or mitigate the risks of those early on in your process development. And that just involves speaking early on between development and supply chain manufacturing.
Yeah, great advice, definitely. Okay, I think we probably have time for one last question. So I suppose sort of looking ahead, will the manufacturing solutions that we've discussed today likely sort of expedite the translation of CAR-T therapies for solid tumors? Or are there some unique challenges for non-liquid cancer immunotherapies that are going to sort of require a whole new bucket of solutions in the manufacturing side of things? Maybe, Greg, you could start us off with this one.
I think the principles that we've put in place for the hematological cancer type CAR-Ts really do apply as far as manufacturing. I think the biological questions are different, and I'm not expert enough in immunology to answer those. But I would say that in general, I don't see major differences. I think we're well positioned from a manufacturing process analytical development point of view to tackle the problems of solid tumors. But I'll let my colleagues speak about it as well.
Great. Chris, did you have any comments on this one?
Yeah, I mean, as Greg mentioned, certainly there is the biological challenges which are very different than that of the liquid tumors, right? I mean, the immunosuppressive microenvironment, I think, is thought to be one of the large barriers. One of the things, I totally agree with Greg that the manufacturing solutions we are putting together now on CAR-T should translate very well, whether it's solid tumor or whether it's liquid tumors. And there may be certain critical qualities that are different in the T cells that we need for that, but the process that we are envisioning should take care of that. One of the, it could happen that for solid tumors, it becomes a sort of multi-treatment regime. So you may have a checkpoint inhibitor going in with a T-cell therapy, and so that has logistical challenges for the companies and the industry who are building that. But I think independently from a manufacturing standpoint, the overall structure, the infrastructure that's being put together can work for solid tumor as well, once we know what are the therapies that are effective for it.
That's very true. Thank you. And then Ian, any last comments?
Yeah, I mean, I agree there's going to be biological challenges. I think the combination therapies, things like checkpoint inhibitors, will definitely have to be looked at. But I do see some changes coming in the manufacture. So mainly because I think the CAR T cell is going to have to be much more sophisticated. So it's not low-hanging fruit like the B-cell leukemia. We've got this tumor microenvironment that we have to bite our way through. We have to get the T cells into those tumors in patients where T cells don't go into the tumors. So I think it's going to be much more complicated. The genetic modifications and manipulations are going to be more complicated. I think there'll possibly be multi-step modifications. So we might need to combine gene editing approaches with vector transduction approaches, maybe doing 2 or 3 modifications serially or in combination to bring in extra factors, allow cytokine production by the T cells, like this truck system, or bringing in chemokine receptors, or masking or knocking out the checkpoint inhibitors on the T cells themselves to allow them to not to be inhibited then in the tumor microenvironment. And another thing that might come into the manufacturing would be possibly the formulation of the product might need to be different depending on the way it's going to be applied. So for some solid tumors, it might be advantageous to directly inject into the tumor site if it's accessible enough. Intracranial injection is already being used for glioma therapy, for example. So if we're looking at closed systems, how do you get your cells into something you can inject, still being in a closed system? Probably you just use a bag, and at the clinic you'd use a septum and take it out through a needle or something. But these things also have to be thought about. So there's going to be a little bit more complexity there. 90% is going to be the same, but I see there's 2 points. One is the genetic modification. Maybe the end phenotype of the cells may have to be different, so the assays would have to be different to make sure they have particular homing characteristics to get into that tumor. So maybe a bit of fine-tuning in the process, but generally 80-90% I think would probably be identical.
Oh, interesting. Okay, thank you. Well, sadly, it does look like we have run out of time today. Very sorry if we didn't manage to get the questions that you submitted. There's been quite a few coming through directed to specific panelists. So what I will do after the webinar today is pass these on to our panelists and see if we can come back to you with a response shortly. But I would just like to say a huge thank you to our panelists today, Ian, Greg and Krish. I've thoroughly enjoyed the conversation today, and I hope our audience members have as well. Thank you for joining us, and I hope to see you again at our next webinar. I will be sending out the recording, the on-demand version, tomorrow, so look out in your inbox for an email from me. And just even say thank you again to our panelists. I'll see you again. Thank you.